Okay, so have you ever thought about how your brain, with its billions and billions of connections, decides which ones to keep and which ones to just toss out? It sounds kind of wild, right? Well, it turns out scientists have just found a huge piece of this puzzle. We're talking about a protein that's basically a personal bodyguard for your most important brain connections, your synapses. So let's dive into this. It's like a spy thriller, but it's all happening inside your head.
All right, so first things first, we got to wrap our heads around this really bizarre but totally essential process that's going on in your brain right now to make it more efficient. And here's the paradox. It's a big one. To actually build a smarter brain, you first have to forget.
Yeah, I know, it sounds completely backwards, doesn't it? How can you get stronger by removing parts? But believe it or not, this is one of the most fundamental rules for building a really high-performance, mature brain.
So, this whole process, it's called synaptic pruning. Picture this. When you're a kid, your brain is it's going wild, making way more connections, synapses, than you could ever possibly need. It's like an overgrown garden. Then the brain comes in like a master gardener and starts carefully trimming away all the extra unused connections. Why? So the really important ones can get all the resources and grow stronger and faster.
But here's the thing. This is a serious balancing act. The brain is walking a razor's edge here. Getting it just right is absolutely crucial. Because if you prune too little, the brain circuits can get all noisy and overexcited. We see that linked to things like epilepsy. But if you go too far and prune too much, you start losing critical connections. And that's implicated in some really devastating disorders like Alzheimer's and schizophrenia.
So, that's the billion-dollar question, right? How in the world does the brain actually do this? What are the tools it's using for this super high-stakes gardening job? Well, the answer is just incredible. It actually comes from a totally different system in your body.
Get this, the brain doesn't even have its own dedicated demolition crew for this job. Nope, it just borrows one from the immune system. It's this whole cascade of proteins called the complement system. Now, normally, its job is to tag invaders like bacteria for destruction. But the brain, being clever, has repurposed it. It now uses that same system to tag synapses that need to go.
Okay, so here's how the demolition works. It's basically a three-step plan. Step 1, a protein called C1q shows up. Think of it as the foreman on the construction site. It finds a synapse that's got to go and slaps the first tag on it. Step 2, that first tag kicks off a chain reaction, which leads to another protein, C3, getting plastered all over the synapse. This is like the crew coming in and spray painting "demolish" in big letters all over the building. And finally, step 3, the brain's cleanup crew, these cells called microglia, they see that C3 spray paint, they know what it means, and they just engulf the whole synapse. Gone.
So this leads to a massive, massive question. If you've got this super powerful demolition crew just patrolling your brain all the time, how do you stop it from wrecking everything? How do you protect the connections you actually need, the important stuff?
Right. There has to be some kind of don't eat me signal, A way for the good synapses to tell that cleanup crew, hey, back off, I'm important. For years, scientists were stumped. They knew this signal had to exist, but they couldn't find it. But they did have a prime suspect. And that suspect, the main person of interest in this whole investigation, is a protein called SRPX2. Now, for the longest time, researchers knew this protein was important. They just had no idea what its actual job was. It was a total mystery. So let's open up the case file on SRPX2. Here's what they knew. 1, it's made by neurons. 2, it seems to be involved in actually creating synapses. And 3, and this is a big one, mutations in the human gene for SRPX2 are linked to things like language disorders and even epilepsy. But the how it did all this, that was a complete black box. And then they got their big break, a real game changer. When they looked at the actual structure of the SRPX2 protein, they noticed something. It had this very specific shape, a motif, called a sushi domain. Now, why does that matter? Well, this sushi domain is like a calling card. It's a structure you almost always find in proteins whose job is to, you guessed it, control the complement system, the demolition crew. This was the clue that just blew the whole case wide open.
Okay, so now they had this critical clue. The chase was on. Researchers started piecing together all the evidence, and they were determined to finally crack this thing.
First up, clue number 1. They ran an experiment to basically see who SRPX2 was hanging out with in the brain. Was it interacting with anyone from the demolition crew? And bingo. They found that SRPX2 physically sticks to C1q. That's the foreman, remember? The one that starts the whole process. They weren't just in the same neighborhood, they were literally bound together. All right, so if SRPX2 is holding on to the foreman, what happens if you get rid of SRPX2? Well, the result is pretty shocking. This chart shows what they found. In normal mice, the level of that demolition spray paint C3 is pretty low. But in mice that were engineered to have no SRPX2 at all, whoa, the amount of C3 goes through the roof. This is a massive hint that SRPX2's whole job is to stop that foreman, C1q, from doing its thing.
But to really, truly prove it, they designed this absolutely brilliant experiment. Okay, let's break down the logic. We know that if you have no SRPX2, you get way too much pruning. And we also know from other studies that if you have no C3, the spray paint, you get way too little pruning. So here's the ultimate question. What happens if you make a mouse that has no SRPX2 times and no C3? It's like a genetic tug-of-war. Who wins?
And here it is, the big reveal. In those mice that were missing both proteins, the winner was clear. They had too little pruning, exactly what you see when you just get rid of C3. This is the smoking gun. It proves, beyond a doubt, that SRPX2's job is to stop the whole demolition cascade from ever starting. Think about it. If you take away the spray paint can, it doesn't matter if the bodyguard is there or not. The building's safe because there's no way to mark it for demolition in the first place.
And just like that, case closed. The mystery is solved. SRPX2 isn't just some random protein hanging around helping build things. No, it has a very specific and incredibly vital job description.
I mean, the researchers themselves said it perfectly in their paper. They wrote, quote, SRPX2 protects synapses against complement-mediated elimination. It couldn't be clearer. It's a bodyguard that directly stops the brain's own demolition crew.
So, the absolute best way to think about this protein is as a bodyguard. That's its job. Healthy neurons make SRPX2 and post it like a guard at their most valuable synapses. So, when that complement demolition crew comes rolling through the neighborhood, SRPX2 is right there to step in, flash a badge, and say, nope, not this one, you need to move along.
So, why does all this matter? Well, this is a huge deal for a few reasons. First, it shows us a brand new way the brain sculpts its own wiring with incredible precision. But maybe more importantly, it could help us understand a heartbreaking mystery. In diseases like Alzheimer's, why are some connections destroyed while others are spared? Well, maybe the vulnerable ones just don't have enough bodyguards on duty. And if that's true, that opens up a completely new and honestly, really exciting path for developing future treatments.
And that leaves us with one final, incredible thought. We've identified the brain's own internal security force. So the big question now is, can we hire more? Can we find a way to boost our brain's natural supply of these bodyguards? Just imagine therapies that could dispatch more SRPX2 to protect our precious synapses from the ravages of disease. It's an amazing possibility, and one that could, quite literally, change everything.